Ladies and gentlemen, welcome to Day 3 of the 10th International Multithematic Medical Congress, Scientific Cyprus. Thank you.
This morning's session is the oral presentation of selected abstracts. I would like to call the chairs of the next session, Dr. Giorgos Lukaidis and Dr. Antonia Sofokleous.
Well, good morning, everybody. The first oral presentation, it's the externally controllable reprogramming of glioblastoma multiforme by radio frequency responsive genetically engineered neural stem cells. And the presenter, it's Dr. Andriani Odysseos from the Translation, Nanomedicine and Nanobiotechnology Lab, EPOS, Glasses Research and Development.
Good morning, everybody. I thank the organizers for giving us the opportunity to present our work. Well, glioblastoma multiforme is the most deathly, most malignant human tumor, a grade 4 glioma according to WHO classification, a rare but very morbid disease, and with the standard of care over 30 years, temozolomide and radiation therapy, we have not seen any improvement. We have distinct phenotypic types, which are characterized by a combination of genetically heterogeneous subclones, mostly attributed to mutations on tyrosine kinase receptors, and several tyrosine kinase protease inhibitors are currently in clinical trials. This intratumoral variability and heterogeneity arises from glioblastoma stem cells or initiating cells, which also form the foci of recurrence. Besides target genes, the blood-brain barrier and the associated blood-brain tumor barrier, comprised of endothelial astrocyte and pericyte structures, prevent delivery of therapeutics and comprise a major failure to therapy. And we manage this with diverse design guidelines for nanograms. A second barrier is the interstitial tissue microglia mechanical barrier, and we manage this with delivery through the pores of tumor interstitial space, the so-called channel propagation. Induced neural stem cells emerge as a multiponent weapon in glioblastoma because of their tumor homing and killing capacity, their nano-drug delivery and actuation potential. They are reprogrammable as drug carriers or biosensors of recurrence and progression, and they can pass through blood-brain barrier and interstitial tissue extracellular matrix, so they have a very strong migrational potential. Radiofrequency genetics is emerging as an arena in cancer therapeutics. Before radiofrequencies 0.9 to 2.45 gigahertz, it used differential gene expression without genotoxicity, the so-called industrial science and medical band. We can use it with no license. And studies regarding mechanism of action, besides thermal effects of radiofrequency, are missing. And there are several methodological flaws in the existing studies. So research for basic understanding and for development of novel therapeutic products under RF transcriptional control is highly sought. All these concepts, are collectively integrated under the concept of externally controllable molecular communication, which emerges as a super-discipline of wireless communication, where we convert biochemical interactions to wireless signals. And our objectives here is an externally controllable system of neural stem cells for glioblastoma, assess transmigrational tumor homing and tumorotropic and tumoricidal potential, generate glioblastoma reprogramming cells, and build the RF interfaces, therefore a complete in-messaging therapeutic loop. The first thing was to select a very potent therapeutic weapon, miRNA-34, which attacks multiple pathways in both glioma cells and the glioblastoma stem cells. And we generally induced neural stem cells from fibroblasts through transdifferentiation. We assess their potential to create exosomes. They are well characterized by nanotracking. They successfully overexpress miRNA-34, several-fold higher than housekeeping genes, and we then had to induce therapeutic miRNA under RF. We chose INGR1 because it modulates fundamental gene networks maintaining low baseline expression levels and is used by modulated radiofrequency radiation fields of 900 megahertz, and its average specific absorption rate, which is the rate by which energy of RF is absorbed, is below the maximum acceptable that of mobile phone range. In order to study in vitro migration potential, we set a triple co-culture in 3-well silicon insert with fibroblasts, neuron stem cells, contrasting U87 glioblastoma cells. And 5 days preceding, we had compromised glioblastoma structural characteristics, apoptotic morphology, and tumorotropis and tumoricidal effect. This was assessed in vivo in animals carrying orthotopically implanted glioblastoma tumors. So what you see red is glioblastoma tumor. And contralaterally, diametrically opposite position, we injected these neural stem cells. So you can perfectly see the migration and the invasion of these cells within the tumor bulk. And this transmigration resulted to significant prolongation of survival in these cohorts of animals. The immediate next task was to create a new system for radiofrequency that could be integrated into routine tissue culture workflows. So, with the help of our engineers, we managed to develop for the first time a bottom-up radiating system which can mount the radiofrequency directly on a standard tissue culture dish. As here you can see the simulation for a 12-well plate, and the distribution of SAR at the bottom of the plate together with the proper medium for these particular cells. So we have several simulations for several tissue culture theories and several culture medium. So we then conducted a study where we expose neural stem cells transfected with the miRNA gene together under the promoter of INGR1 with a red fluorescent dye MK2. And what we can nicely see, if we contrast the thresholded fluorescence intensity to the overall cell-covered area, we have a very, very important finding we shared for the first time. The significant fluorescence increase peaked at 20 hours post the radiofrequency pulse, which was for very, very short time, 30 minutes, under the simulated parameters we described before, whereas most studies use exposure for 24 hours or even days. So we saw significant fluorescence increase peaked at 20 hours post-RF pulse independent of cell proliferation. So we have a highly promising finding suggestive of suitability of ERG1 promoter for interfacing in vivo gene therapy to external control by RF wireless communication, and high promise in hyper-fractionated RF pulses for therapeutic applications. So we can speak of the very first proof of concept for an externally controllable cell-enabled gene therapy. In conclusion, fibroblast-derived neural stem cells serve as glioblastoma therapeutics with strong transmigrational and humoricidal potential in vitro and in vivo. Ultra-short, high-power RF pulses modulate effectively therapeutic mRNA expression in neural stem cells. Interfacing neural stem cells with the mRNA-34 targets via RF-sensitive promotion emerges as a high promise for glioblastoma therapeutics. So we have next-generation theranostics for brain pathologies with nano-communication interfaces. And next-generation scientists seems to be very eager to take up and expand this new technology. This is from Researchers' Night. And of course, no man is an island. This work needs transdisciplinary effort. And I would like to thank all my colleagues at EPOCS in the University of Cyprus, Dr. Prokopi for helping us with nano-tracking, and of course, our funding agencies, European Innovation Council and RIF here in Cyprus. Thank you very much for being here this early morning, Saturday morning.
Thank you very much. Do you have any questions? Okay. Thank you.
We can proceed with the next talk, which will be delivered by Dr. Odiadis from the Center of Excellence in Biobanking and Biomedical Research, Molecular Medicine Research Center, and University of Cyprus Medical School, Nicosia, Cyprus. The title of his talk is Repurposing Chemical Chaperones to the Rescue of Mouse Models of Alport Syndrome.
Good morning. Thank you for the kind introduction. Thank you for inviting us to present our work. I come from Professor Konstantinos Zelta's lab, and I am going to talk to you about our studies, on preclinical studies on Alport syndrome mice treated with chemical chaperones. So Alport syndrome is a glomerular nephropathy characterized by hematuria and proteinuria progressing to end-stage renal disease, and is associated with hearing and ocular abnormalities. It is characterized by clinical and genetic heterogeneity. There is no radical cure at this moment for the Alport syndrome, and until now, ACE inhibitors are used in order to delay the onset of end-stage renal disease. All cases of Alport syndrome arise from mutations in genes encoding the alpha chains of type 4 collagen, the alpha 3, alpha 4, and alpha 5 chain. Despite the many potential combinations, the 6 collagen chains of type 4 form only 3 sets of protomers when we focus on the alpha 3, alpha 4, and alpha 5 protomer. In mature glomeruli, the glomerular basement membrane is mainly composed of the alpha 3, alpha 4, and alpha 5 chains. And in humans, these chains are also present in the tubular basement membranes and the distal tubulars. In mice, in addition to the glomerulus, we have detection in proximal and distal tubules. Many years ago, many knockout models have been created in order to study the Alport syndrome, and these knockout models have a severe Alport phenotype. However, about half of Alport patients have missense mutations with single amino acid substitutions, with glycine being the most common. The first Nogin Col43 mouse have been created in Professor Konstantinos Zelta's lab in 2014, and in order to recapitulate the phenotype of the heterozygous mutation at 1334 from glycine to glutamate in humans, which causes the thin basement membrane nephropathy. The mutant mice showed ER stress in the kidneys, and the mutation we studied has a pathogenic effect, and it is endemic in Cyprus. Also, heterozygous carriers of this mutation range from isolated microscopic hematuria to end-stage virulent disease. So our objectives was firstly to characterize the kidney phenotype of the two Alport mouse models, and next to treat the Alport mouse models with synthetic chaperones in an attempt to rescue or delay the disease phenotype. So we began the characterization of the Noggin mouse that carries the point mutation at 1332 from glycine to glutamate in homozygosity. And the second mouse model was a compound heterozygous model that carries the same point mutation in compound heterozygosity with a knockout allele. Both mutant mice had a shortened lifespan, increased proteinuria, intermittent hematuria, and also increased serum urea and creatinine. Electron microscopy studies showed the characteristic alternate thinning and thickening of the glomerulus basement membrane. This is one of the main characteristics of Alport patients. And also, we found that the expression of the mutated COL4A3 and also of the wild-type alpha-4 and alpha-5 were reduced in the tubular basement membrane in the mutant mice. Additionally, renal fibrosis was detected in kidney sections of the two mouse models.